Hey everybody, today you're going to learn about phylogeny, speciation, extinction, and the history of life on Earth. So this is a picture of the Galapagos finches, and remember when we talked about how their beak shapes were different because of their food. So, some vocab. So phylogenetic trees and cladograms show evolutionary relationships. They use these terms interchangeably. So phylogenetic tree and cladogram, I looked up to see if I could figure out the difference because I always thought they were the same. And they are the same, they're different, so you can just think of them as the same. A clade is a group of organisms that share a common ancestor. So if you look down here, right, this is considered a clade, and this is considered a clade. However, these are not because they don't include, yes, they have a common ancestor here, but they don't include the other ones. Same thing with this. Yes, all of these have a common ancestor, but it doesn't include this. So maybe draw a picture of that so that you know what a clade is. An outgroup would be like, for example, this purple circled thing. And it's distantly related to the group of organisms that you have the cladogram. And what it does is it serves as a comparison to show that these can be measured off of. And then a derived character is a trait that evolved, for example, jaws. So this would be a derived character. So it evolved, and then everything above it has jaws. And claws or nails. So all of these have claws or nails. Feathers, this would be a derived character that only the pigeons have. Okay. All right, so there's different parts for these trees. A node is the most recent common ancestor. So right here, the root is a common ancestor to the whole cladogram. And they can be drawn sideways, they can be drawn vertical. You might also see some things where they can rotate around the node. So like this cladogram could rotate it around this node right here, and it could go E, B, B, or CBA. So here is another example of a cladogram. This is something you might see on an AP test. So down here you have the number of amino acid differences in cytochrome C, which is an important protein. And so you can see between E. ferris and D. polylepis, maybe, there are 21 differences, whereas between E. ferris and E. africanus, there is only one. And so that's why these two are so close to each other, where E. ferris and E. Or D. poly whatever is way over here, right? And so they're the furthest apart. So the number of amino acids is going to show you where you would draw them. So you could get a question that you need to draw the tree based on a table. So this is biochemical data, for example, amino acid differences or DNA, and they are better at showing relationships than morphological or structural data. Okay, so here's one for you to try. So you're going to construct a cladogram on this template, and you're going to use the differences in the list protein, okay, which is just this protein, and you're going to circle the position on the cladogram that represents the outgroup. So go ahead and pause the video and try to do that. Okay, so here is what you should have drawn. So the brown and black were super close together, very few differences. There were 7 differences between the black and the polar bear. And then the panda is the outgroup because it was the one that was the most different. So the next thing we're going to talk about is speciation. So just like Darwin's finches, there are birds in Hawaii on those islands, and they're called honey creepers. And remember the founder effect, where one founder species gets there, and then it differentiates and speciates into all these different ones because of environmental pressures like food. So speciation occurs when populations are isolated reproductively. So when two populations can't reproduce and make fertile offspring, so fertile offspring means that they can reproduce themselves. And this is caused by increases in genetic diversity. So if the two populations get super diverged, then they're not going to be able to make fertile offspring. So some vocab here. So reproductive isolation is going to prevent gene flow between populations. So you've got prezygotic barriers, so that means that the eggs are not able to be fertilized, and there's different ways that can happen, all isolation. So they could live in different locations, which would be habitat isolation. They could mate at different times of the year or different times of the day, which is temporal. Like if one population mated at night and one during the day. Behavioral isolation, this is like bird calls or some kind of mating dance. Mechanical isolation, this is where the sperm and egg can't actually get to each other. There's an example of snails, where snails are twisted one way and then another population, because of a mutation, twisted the other way, and so the egg and sperm can't get to each other. And then the last one is gamete isolation, and that's when the sperm of one species can't fertilize another one. The other thing that can happen to prevent gene flow and cause speciation is post-zygotic. So post means after. Zygote is when the egg and sperm come. So this is preventing the zygote from becoming viable or living and fertile. So you can have a couple things here. You can have hybrid inviability, which means that the zygote just doesn't develop. So the egg and sperm come together, they make a zygote, but then it just doesn't develop. You can have hybrid sterility, and that's the example of a mule. So a horse and a donkey can mate, and they make a mule, but the mule is sterile, can't make its own babies, it's infertile. And the last one is hybrid breakdown. So this happens a lot on rice. And so the first generation is good, but then after they reproduce, then they get weaker and weaker and they're not able to reproduce. So over time, those hybrids break down. So there's different types of speciation. So in order to have speciation, you need isolation, either post or prezygotic isolation. But it can also, but this is another way to kind of categorize it. So you can have allopatric speciation. This is the most common, where it's divided by a geographic barrier. So like a river right here, right? Or a canyon or an island, right? So here you have a river, and eventually it's going to prevent the gene flow. These are going to change based on the environmental pressures, and these are going to change, and then pretty soon, if that river was to go away, they'd be so different they wouldn't reproduce. So allopatric is when they're geographically isolated. Sympatric is where it happens in the same area. So this is an example of sympatric. And so they speciate even though they live in the same area. And this can result from mutations, polyploidy, where instead of 2N, an offspring has 3N. And it's a new species. It can't mate with the parent population. So that would be this first one. This happens in flowering plants. Habitat differentiation. I'm going to show you a picture of some fish, and they live in different habitats in the lake, and they have speciated, or sexual selection. So this is an example of the sympatric speciation in African cichlids, which is a type of fish. And the species in each lake are descended from a single ancestor, right? And so there's no barriers within the lake, but there's separation because of the habitat. And so that would be an example of sympatric speciation. So you get different species in the same location because of different habitats. Okay, so evolutionary trees, there are 3 types that you need to know. And I would go ahead and pause the video and draw these. Okay, so gradualism, just like it sounds, it's a gradual changes. So many morphological, so that's form, changes accumulate over a long period of time. So really small changes happening over a long period of time. Um, Punctuated is this one. So this one is a quick change, right, after long periods of no change or stasis. So it's no change, a quick change, and then again, no change, right? And then adaptive radiation is this one, and that is where it's really now fast in terms of evolution is, you know, thousands and thousands of years. But a niche is opened up, and that allows there to be habitat and different organisms can come in there and they radiate. So Darwin's finches is an example of this. Mammals are an example, which I'll talk about in a minute. And so it's where a single lineage gives rise to a bunch of different species. So here's our example of mammals, right? So here's our ancestral mammals, and then they became all these different mammals we know of today. And if you think about why that happened, well, it happened right around the time that the dinosaurs all went extinct. So these ancestral mammals lived underground, and they survived the meteor that hit and made all the dinosaurs go extinct. So since there weren't any dinosaurs to compete with or eat them, they were able to move above ground, and they have all this new habitat and niches, and so they were able to diversify pretty quickly. Okay, so extinction. So they play a major role in Earth's history. There's been 20 or more large-scale extinctions, one of them being the dinosaurs. And what happens is after the extinction, there's reduced diversity, and then it's followed by adaptive radiation, where there's quick speciation because of the open niches. Small scale extinctions, like if we're talking about populations, are likely to happen if there's low diversity, genetic diversity in the population. So an example of that is the California condor, ugly creature who's kind of cute. So in 1987, it became extinct in the wild because of habitat loss, lead poisoning, and poaching. The last 27 were captured and they were bred in captivity, and then they were reintroduced. And they found that all of the California condors are descendants, the ones that now, of 14 condors that were bred in captivity. So really, really low diversity. So here's an image. Here's your California condor. Whoops. Back. Here's your California condor. Different ones within it with different, genetic variation, and then a huge amount of habitat loss, poaching, and lead poisoning led to only a few surviving, and then their offspring are the result of them, and so there's a really low diversity. So if a disease was to come through or something, right, there wouldn't be a lot of variation, and either all of them would survive or none of them would survive. So that's why populations with low diversity tend to go extinct easier than populations with high diversity. So this is something that you just need to know as a biology student and as a human. And that's how we name things. So each species has a two-part Latin name. The first is the genus, and that's more general. And the second is specific, and that's why it's called species. So our species name is Homo sapiens, or H. sapiens. And it's important, a couple things to notice is it's italicized. The second word is not capitalized. And if you want to do it shorthand, you can just do H for the genus and then sapiens. Now, if you can't italicize something when you're writing it, you would just underline it. Okay? All right. So, 6 kingdom versus the 3 domain. So this is how we group organisms in the world. So the kingdoms are right here. So you have bacteria, you have archaea bacteria, protists, which is like the paramecium and amoeba, plants, fungi, and animalia. And then another way to group it is based on these 3 domains. So you have eubacteria, and that would be like the bacteria we always think of, like Salmonella and E. coli and all the ones that make you sick. Archaebacteria are the ones that live in like the hydrothermal vents in extreme places. So they're thermophiles, which love heat, that live down in or in the geysers in um, Montana. And then eukaryotes, which we know are eukaryotes. And what I find is super interesting is that with this domain, 3-domain chart, and they've done this from DNA evidence in the last 20 or so years, that it is shown that those two types of bacteria, even though their bacteria are as different from each other as we are from them, which is crazy. Like one bacteria is as different, DNA speaking, to E. coli than we are to E. coli. And we're more closely related to these archaebacteria than we are to the bacteria. So, okay, last topic on this one is the history of the world and the atmosphere and how things came to be. So Earth was formed 4.6 billion years ago. They've done dating of rocks, so they know that to be true. And the first fossils that they have evidence of was 3.5 billion years ago. So the atmosphere on early Earth had hydrogen gas, so H2, N2, carbon monoxide, CO, carbon dioxide, CO2, but there was no gaseous oxygen. So there was no O2 in the atmosphere. So we couldn't have survived. They also, there's a hypothesis that the first genetic material may have been RNA. And the reason for that, the evidence is that RNA can assemble spontaneously. So they've done experiments and RNA can just put itself together. And but they don't know how the switch of RNA to DNA might have occurred. And then how organisms, eukaryotes came to be. We've talked about this before, the theory of endosymbiosis. So if you think of, we know that mitochondria and chloroplasts were once their own unique little free living prokaryote, right? And they were engulfed by a larger prokaryote, and they became symbionts, right? They lived inside of them and were helpful. And some evidence we talked about is they both have DNA, their own DNA, and it's circular, like bacteria have. They can reproduce on their own. They have ribosomes. They can make their own energy. Okay, so all these things are the evidence for endosymbiosis. Okay, so here's an example problem. So here's two different experiments, and they're testing how organic monomers maybe came to be. So it says two groups of students attempted to recreate the primitive atmospheric conditions of early Earth using the apparatus represented in Figure 1. Each group ran the experiment with different gas mixtures in the apparatus. Predict which experiment has the greatest probability of generating amino acids and nucleotides. So they've done experiments like this where they put the atmospheric gases from early Earth inside of a vacuum, right? And then they charge it with electricity because they know that the Earth's atmosphere had electricity. And they've actually discovered that that monomers have come to be, right? They've just having those compounds and different monomers like amino acids and nucleotides have formed. And so if that can happen in several weeks or a month, then it's very likely that, amino acids can get put together to be proteins, and nucleotides or assemble themselves into RNA and DNA. But anyway, so go ahead and pause the video and look and see if you can answer this question. Okay, so the answer, experiment 1 will be able to generate amino acids because of the nitrogen. Remember, nitrogen is a requirement So amino acids have nitrogen, carbon, oxygen, and some sulfur. And then if you look at nucleotides, neither of the experiments would be able to produce nucleotides because they require phosphorus. And none of these gases have phosphorus. All right, and that's it. I know that was a long one. I hope you learned some about speciation and extinction and early life on Earth. And we'll see you next time.